Hi, everyone, and welcome to our Health Professions Week event for Pcat. My name is Justine. I'm going to be walking us through some great strategies that you can use as you prepare for your future career in pharmacy. I do want to say that I am joined by one of my fantastic colleagues who will be working behind the scenes to answer any questions or concerns that you have. His name is Paul, so if you have any questions, you can certainly drop us a line in that Q&A and we'll get back to you. So don't ever feel bad asking questions. That's really why we're here. We want to make sure that you are well prepared for your future career. So in our event, we're going to just talk about the Pcat because I want to make sure that all of you have a good idea of the structure and format of the test, since there have been some changes over the past couple years. We'll look at how you should be approaching questions on the test, and this is a strategy really that you can use for all multiple choice tests. The Pcat, any tests you have in college, and the licensing exams you'll take as a professional to get your career really started. And of course, since this is boot camp, we will be going through some exercises specifically in the sciences, and we'll answer any questions that you have as we go through our event. The other thing I do want to say is this is how we love to do our classes, with teacher on camera and another fabulous instructor behind the scenes. So make sure to get all of your questions answered. I cannot say that too many times. If you have questions, definitely reach out and let us know. Now, looking at the structure of the Pcat, you're going to start with a writing prompt. This will always have the same general format, asking you to just give some solutions to a world problem. There's only one prompt that you have to choose from, but you're not expected to be some sort of like super expert in like world events. Just being aware of just general events and being able to relate your own experiences will serve you very well. So it's not about having like the most brilliant solutions ever. It's just being able to organize your thoughts in a concise way and backing up your solutions with evidence. I definitely would recommend writing several essays before you sit for your exam, just to get more comfortable with writing under a time constraint. The next two sections will be your biology and chemistry sections. These have changed in the last couple of years, mainly that they are now passage-based. So about 50% of the questions in both of these sections will be associated with passages. Each passage will be about 200 to 300 words and will be associated with 4 questions. So that really tests your ability to integrate new information with your own content knowledge and think critically to answer questions. So it's definitely a lot more problem-solving based than it has been in previous years. You will have access to a calculator. It will be a 4-function calculator, so not the best calculator, but you do have that advantage over previous years. Then you'll take a 15-minute break. I highly recommend that you get up and walk around during that break, get some snacks, get yourself refocused, because you still have 2 more sections. A critical reading section, which is solely passage-based. You'll have 6 passages, each with 8 associated questions, and this can come from both natural sciences and humanities. So I would recommend reading not just science. I would get started into more like social sciences type passages, like things from like The Economist. So kind of like the not as exciting stuff to read if you are a science major. The last part of the test will be quantitative reasoning. This will cover all the way from basic math all through calculus. It is much more word-based problems than in the past, so about 50% of the questions will be word problems. So be sure to do adequate practice. That's the #1 thing that I see make a difference in the success of students is really not just studying content, but also giving yourself adequate practice. It's practice that makes permanent and practice that makes proficient. And just studying content alone won't get you the whole way there. Well, that's why we're here, learning strategy and reviewing problems. So when it comes to working through multiple choice tests, you definitely want to be strategic. One of the big pitfalls that I know I made as a student, and hopefully we can help you avoid making in the future, is anytime I had a multiple choice test, I felt like I would read the question and then look immediately at the answers and kind of skip through that thinking about the question stage. So you do want to make sure that you give yourself an opportunity to think more about what the question is asking and also what information you need in order to solve the question. You also don't need to do the questions within a particular section in order. So the first thing that you want to do, and this goes for every section of the Pcat, is to first stop and characterize the answer choices. This is to give yourself an opportunity to determine the level of difficulty of the question and also the amount of time that it's going to take you to answer that question. Because based on the pacing, depending on the section, you'll have anywhere between 30 seconds and about a minute to answer each question. If you have something that's really calculation-based, it's not really in your best interest to start with that question. That's why one of our main strategies that we teach is called triaging. It's making the decision to save a more time-intensive, more challenging question for a second pass through the section. So you definitely want to pick an answer. Don't ever leave anything blank on the exam. Pick an answer and then come back to it later. And that will allow you to answer as many questions as possible in the allotted time that you have. The other thing I want to point out, as Petros has mentioned, is we do have a drawing for a free Pcat prep book, so definitely enter into that. The one thing I will say anytime you're prepping for one of these exams, and I'm really glad Petros mentioned this, is that you don't want to use your college books. So you always want to use a Pcat-specific reference, mainly because I always say the Pcat is like a mile wide and an inch deep. Most students will spend about 200 hours to 250 hours studying for this test. So you want to make sure you're using your prep time effectively. So don't study stuff that's not within the scope of the exam. Now, once you decide to do a question, you're going to think, and this is where you identify the real task of the question. There's nothing more frustrating than getting a question wrong because you're answering the wrong question. So it can help to just paraphrase the question in your brain. Sometimes it can even help to just jot down on your scratch paper what you're looking for. And then you're going to start to plan your attack. What information do you need from the question stem, from the passage, from your own content knowledge? And from there, this is the real kicker of the strategy. You make a prediction. You go into the answer choice with an idea of what you are looking for. This is just like going to the grocery store with a shopping list. You're going to be much less likely to pick the garbage compared to what you actually came there for. And then armed with your prediction, you match. You look at the answer choices to find what truly matches your prediction. And going through questions in this manner will slow you down at first because you're actually starting to pay more attention to what you're doing. You're not getting that panicked rush through the exam. But you'll find as you start working questions more methodically, you're going to get faster at it and it's going to improve your accuracy. So definitely be strategic in how you answer your questions. And we're going to focus on applying this strategy as we go through some different content areas. So we're going to look at some stuff from biology. We'll also take a quick peek at some chemistry. Now, the biology section, will cover general biology. That would just be kind of like your Bio 101 course from college, anatomy and physiology. It's much more focused on the physiology aspect of things. So you don't need to like memorize all the cranial nerves or anything like that from anatomy, but it really focuses on structure function relationships with organ systems. And then you want to know some microbio. If you haven't taken a college microbio class, that's totally fine. The microbio really just tests the four main types of pathogens. You have the viruses, parasites, bacteria, and of course, the party guys, the fungi. So if you're aware of the general life cycles of these microorganisms and different mechanisms for treatment, you're going to be in a really good place for your exam. Now, in terms of timing, you'll have 45 minutes to answer 48 questions. Remember, half of those questions will be passage-based. So if you account for the amount of time it takes you to read and map your passages, you're looking at around 40-ish seconds per question. Now, of these 48 questions, 8 are experimental. They won't count, but you'll never know which ones are experimental. Now, that can be frustrating, but you can also use that to your advantage. Because if you see a question where you're like, oh my goodness, I'm not sure what to do with this, you know what? You can always guess, pick an answer, and say, oh, that was probably experimental. So it's a way that you can actually use to manage your stress on the exam. Now, if we break down these content areas a little bit more, we can break down just general bio into health, which does relate into anatomy and physiology a bit. So you're looking at like diseases, both on a micro and macro scale. Cell bio, which is essentially like knowing your organelles, and general metabolism. And then diversity of life, which would encompass genetics, which is what we're going to talk about today. So what I really want to focus on is pedigree analysis, because pedigrees are actually pretty high yield on the exam, mainly because they allow you to be tested on both probability, since you can look at the likelihood of inheriting certain diseases. They can test also on inheritability based on different modes of inheritance. So we're going to look at 3 different types of inheritance that are tested on the Pcat, and also what to look for specifically in a pedigree to identify a particular modality of inheritance. Now, we have this little chat box, so as we go, I know that you all can ask questions, but I'm also going to ask some questions of you as well, just to get you thinking a little bit more about the material. So I always say, if you want to learn, the best way to do so is to really engage yourself in the material. So we're going to start with autosomal recessive inheritance. And go ahead and tell me, or if you're a little bit shy, you can always just think it to yourself. You know, talk to your computer a little bit. How do we prove recessive inheritance? There's actually 2 different things that we can look for in a pedigree. And while you think about that, I'm just going to give you a little background about a pedigree. So pedigrees are ways of tracing a particular trait through a family tree. The circles in pedigree are females. Always remember that, since women have curves. The males are squares. Anyone who is affected or has a particular trait of interest is noted by a filled-in circle or square. And anyone who is not affected, does not have that trait, will be a blank circle or square. So anytime we're looking to prove a mode of inheritance, what we want to look for are 2 parents that have the same phenotype that have an offspring with a different phenotype. So in autosomal recessive, what we are looking for is this pattern, where we have 2 parents that are both unaffected that have affected offspring. Another way to say this is we're looking for generational skipping. In this case, we know that both parents must be carriers of the trait. Remember, a carrier is somebody who has the allele but does not show the associated phenotype. We can only have carriers for recessive inheritances. You do not see carriers for dominant traits. So that's recessive. Look for 2 unaffected parents with affected offspring. We also have dominant inheritance, and this one tends to get a little bit trickier. A lot of students always say that you can prove dominant inheritance based on the fact that a trait appears in every generation. Now, that certainly is true of a dominant inheritance, but a lack of generational skipping does not prove dominance. In fact, if you have a family that just has a very high instance of a recessive trait, you certainly can see that trait appear in every generation. So we're not looking for, black circles or squares in every generation here. What we're looking for is something that cannot happen in a recessive inheritance. If we have recessivity, 2 affected parents can only have affected offspring. So we could not see something like this in a recessive inheritance. If it was recessive, 2 affected parents could only make affected offspring. So by seeing 2 affected parents have unaffected offspring, we can say this is certainly not a recessive inheritance, which makes it dominant. So this is the proof that we look for recessivity, for dominance. We cannot just look at the fact that this trait appears in every generation. So your proofs will always be specific matings between parents. You're always looking for matings between parents with the same phenotype that have offspring with a different phenotype. The last type of inheritance that you can get tested on is X-linked recessive. This is the same thing as saying a sex-linked trait. You're not going to see Y-linked traits on the Pcat. For one, they are very rare and they're not really tested. The same thing goes for X-linked dominant traits, just because it's very hard to distinguish that from a general dominant inheritance. So really, these are the 3 types. Now, when we look at an X-linked recessive inheritance, one thing that you may be thinking looking at this pedigree is, wow, there are certainly a lot of affected males, and that is characteristic of an X-linked trait. When it comes to females, females have 2 copies of the X chromosome, so we need 2 copies of the recessive allele in order to show the phenotype. When it comes to males, males only have 1 X chromosome, and it's actually different genes encoded on the X and Y chromosomes. So males only need 1 copy of the recessive allele in order to show the phenotype. And that's why if you ask around, you're more likely to find a lot more colorblind males than colorblind females, since your photoreceptors are carried on your X chromosome. So just to recap, to prove X-linked recessive, you're going to still see that same pattern of recessivity where you have 2 unaffected parents with affected offspring. The main differentiating factor here, though, will be that most of your affected individuals will be male. So what we're going to do here is work through some practice questions together, really focusing on how we can be strategic in how we answer these questions. So we have 4 questions, or 5 questions that we're going to work through. And the first thing that we're going to do, as always, is to stop and characterize our answer choices. Here our answer choices are percentages, and this would be one of those probability questions that we can see associated with pedigree analysis. Now, the next thing we need to do is think, and this is where we want to organize the information provided to us and also determine the task of the question. Now, one thing I noticed about this question is it is asking for the likelihood of the offspring being a carrier. So to me, that would tell me that this has to be a recessive inheritance, because that's the only way you can have carriers. The other thing we notice about this is we actually are given a pedigree. And go ahead and tell me in that chat, what do we know about our offspring in terms of his phenotype? Because this is the key for answering this question, is making sure we use all of the information that is available. We're looking for a carrier. It's a recessive inheritance. The other thing we know is that the square is not filled in. This individual is not affected. So that's something we're going to need to bring into our analysis of this question. So let's think about the possibilities of genotypes for this individual. And we can do this with a Punnett square, where we're going to put the genotypes of the parents across the top and side, and this will allow us to determine the possible genotypes of the offspring. So we can just fill in our Punnett square. And these are the 4 possible genotypes that the parents could produce. This is the common 1 to 2 to 1 ratio you may be familiar with when it comes to monohybrid crosses. Now, the key here is noting that our individual was not affected. This actually allows us to eliminate, as a possibility, being homozygous recessive. We know that we're not r because our person, our male in question, is not affected. So they cannot have the genotype of r. We know that based on their phenotype. From here, we can see that we have 3 possible genotypes remaining. Just based on an unaffected phenotype, we cannot distinguish between homozygous, dominant, and heterozygous. So all 3 of these will remain possibilities. From there, we can just say there's 2 different ways, 2 different combinations that give us a carrier out of 3 total genotypic possibilities. That would be a 2/3 probability. So this would be the type of question that would certainly be a little bit higher difficulty, but notice that taking a strategic approach and really taking the time to think about the information provided, we're able to answer this question efficiently and accurately. And that's really what we're going for on this exam. So we're going to go through a few more questions. And if you want, you can certainly type in your answer into this chat as we go, to get yourself some practice as we work through some questions. Now, for this next one, we certainly can tell that we have a large pedigree, and all of our answer choices are different modes of inheritance. So we see those 3 we talked about, autosomal recessive and dominant, along with X-linked, and then there's that cannot be determined. Now, I personally hate that as an answer choice because you always feel like they're going to give you enough information, but sometimes they won't. So we're going to go through that same sort of decision tree, that same sort of critical analysis, and we're going to look at a couple different pedigrees to really emphasize those proofs that we had mentioned before. So what we have here in this pedigree are equal numbers of affected males and females. That alone will give you sufficient information to rule out X-linked recessive. If it's X-linked, you're pretty much going to see like only affected males, maybe like 1 affected female, but it's going to be like mainly guys. Now, when it comes to autosomal recessive versus dominant, this is where we need to look for that proof. Remember, proof was 2 parents with the same phenotype having an offspring with a different phenotype. We have 2 different matings in this pedigree. We have the grandparents, which are affected and unaffected, and then another couple on the left side, again, affected and unaffected. These types of, I don't know, like a like a technical way to say this, but like matings of mixed phenotypes are not diagnostic for identifying a mode of inheritance. So in this case, we actually do not have sufficient information to distinguish between dominant and recessive. So in this case, we actually cannot make a determination. So in the absence of proof, you have to go with cannot be determined. And if you wanted to, you could absolutely go through and with this pedigree, write out genotypes that work for both recessive and dominant inheritance. So in the absence of proof, we don't have sufficient information. Now let's take a look at a different pedigree. Notice the question stem for this one is pretty much identical. We're just swapping out a different pedigree. So again, we're going to look for one of those proofs of inheritance, looking at a mating between parents with the same phenotype having offspring with a different phenotype. In this case, we actually see that right here in red. We have two unaffected parents with affected offspring. This is sufficient proof of recessivity. We know that both of these parents have to be carriers, so it's got to be a recessive inheritance, and we see a roughly equal number of affected males and females, and that rules out X-linked. So this would have to be just straight up normal autosomal recessive inheritance. So we would be able to confidently match answer choice A and keep going. So by making our predictions, we're able to quickly select an answer. So we spend a little bit more time up front in the analysis, but we're able to make fast decisions in the answer choices. So if you're the kind of person who tends to waffle a little bit between answers and try to like talk yourself into like different answers, making a prediction will really help you. Now we're going to move away from specific pedigrees and look more into genetic testing. So here we have a pedigree, and the question stem actually tells us the mode of inheritance, but honestly, even if it didn't, we should still be able to identify this as X-linked recessive because we have just mainly affected males. Now, in our answer choices, we're looking at males versus females. Now, looking at our question stem, this is telling us that our individuals in generation 4, denoted by the question marks, are going to have children of their own, but they're worried about their offspring inheriting an X-linked recessive trait. We want to know who should undergo DNA-based genetic testing. Well, let's think about this. What do we use DNA-based genetic testing to identify? Well, looking at these individuals, you'll be able to just see their phenotype. So if you have someone that is affected who has this particular trait, you will be able to see it. So we can identify individuals that are homozygous recessive. We can identify people who are affected. Now, we use DNA-based testing to identify carriers, those who have the allele but do not show the trait. Now, in an X-linked recessive inheritance, we can have females as carriers, but males cannot be carriers. They only have one copy of that X chromosome, so they're either affected or they are not affected. So for this reason, there's no reason to test the male. Just by looking at him, we can identify whether they have the normal allele or if they have the affected allele. The female is another story. We know that that females in the first generation and the second generation are carriers based on the fact that they have affected sons. We don't know if the female in the third generation is a carrier. She may be, she may not be, and she may or may not pass that trait down to her daughter. So there is no way of knowing if the daughter in the fourth generation is a carrier. So that's the one that we would need to test. So only the female needs to be tested, because only females can be carriers for X-linked recessive traits. Now, the last one we're going to look at here is going back to doing some percentages. And I do want to point out, if you have any questions about any of these problems we've worked through, definitely let us know, because I know Paul's always happy to answer questions. I'm happy to answer questions. So don't be afraid to reach out if you have, if there's anything that we've looked at so far that was challenging or confusing in any way. Because really, in our classes, we do want we want it to be a safe place for making mistakes, because it's much better off, you're much better at making mistakes in practice than you are making mistakes on test day. So that's my motto to live by. This last one, we're looking at the likelihood of a male in generation 4 having the recessive phenotype, essentially that individual denoted by a question mark box. And our answer choices are percentages. So we're looking at a probability. Now, if we look at this particular pedigree, we see a lot of individuals that are affected, but also note the mating that this individual is a product of is from 2 parents that are unaffected that have already had an affected offspring. So this would be a great example of a recessive pedigree where the trait appears in every generation. So we know that this is recessive, and we know that both of the parents have to be carriers. So we can make that same Punnett square as before, considering the different possible genotypic outcomes. And we're looking at having the recessive phenotype. So we have to be homozygous recessive. We have to have a genotype of little r. The other thing to consider is we know that the other child was affected. However, each fertilization event to create a new person is an independent event. So even if you've had, if there's a family that's had 5 affected children, the probability of the 6th one being affected is not in any way influenced by the previous children. So in this case, even though there is an affected child already, the probability is still the same. There's 4 possible genotypes. Only 1 is affected. So we're still looking at a 1 in 4 probability. So the key takeaway here is each fertilization event is an independent event. The probabilities are not affected by previous outcomes. So if you have questions from this, definitely reach out to us in that chat. The next thing we're going to take a look at is the chemical processes section. So the chemical processes is pretty much identical in format to the biological processes. So again, it's 50% passage-based. These passages can be both informative and experimental. So it actually is pretty fun. I love the passages. So looking at the content, we're going to see roughly 50% from general chemistry. This will be pretty much Gen Chem 1 and 2 in college. So it covers all the way from atomic structure to equilibrium and acid-base equilibrium. You'll also see organic chemistry. This tends to focus more on organic chemistry 1 from college. So you'll go through just properties of organic compounds, different types of isomers, and a lot of nucleophilic substitution reactions. So SN1, SN2, E1 and E2, nucleophilic acyl substitution, and general reactions of functional groups. You don't see as much organic chemistry 2 tested, which is really heavily into the more obscure reactions. The other 20% is from biochemistry. Now, this does not mean in any way that you need to like memorize the citric acid cycle or any sort of craziness like that. In fact, the biochem is more what you will have seen in general biology. So if you're feeling cool with like central dogma, DNA replication, transcription, translation, mutations, just properties of nucleic acids, proteins, and fats and fatty acids, you know enough biochem to be successful on the Pcat. So again, mile wide, inch deep. Now, if we break down the orgo a little bit more, we'll see both properties and reactions. Now, one thing that I see a lot of my students struggle with, particularly with organic chemistry, is reactions. Mainly because a lot of us have been taught organic chemistry as a, hey, I need you to memorize this long list of reactions, and if you can just regurgitate these from memory on an exam, that'd be great. Now, the issue with studying organic chemistry in this way is that you kind of miss the big picture of organic chemistry, understanding why things react the way that they do. So when you study organic chemistry and when we teach organic chemistry in our classes, we don't just focus on like memorize this list of reactions. What we like to focus on more are some of the general properties that underlie why things react the way that they do. So even if you see a reaction on the exam that you may not be familiar with, you can still analyze the reactions, the reactants, and the reaction conditions to predict what the product would be. So for this reason, what we really wanted to focus on was some of the general players in organic reactions, and then how those relate to differences in reactivity between SN1 and SN2 reactions. So here are our general players. So we have nucleophiles. Now, a lot of what we see in terms of nomenclature in organic chemistry is very formulaic. That goes for just IUPAC in general, which just is a giant rulebook, and also the way in which we've named a reactant. So if you think about the word phile, loving, a nucleophile is something that loves the nucleus. So nucleophiles seek out positive charge. And if you think about what I call the Paula Abdul rule of chemistry, opposites attract. So our nucleophiles are electron-dense species. And these can be either strong nucleophiles, things that have full negative charges, things like hydroxide, chloride, negatively charged species, or they can be weaker where we have only partial charges, things like water and ammonia. Where we have lone pairs, we don't have that full negative charge. You can also see pi bonds working as nucleophiles as well, mainly because the pi bond has a lot of electrons concentrated in one area between two atoms. So those are more reactive bonds. We also have electrophiles. Well, if a nucleophile loves the nucleus, an electrophile loves electrons. So electrophiles are electron deficient species, and these will have positive charges. We can have awesome electrophiles like the carbocation, a carbon with a full positive charge. Good old carbocation is the best electrophile that we have in organic chemistry. Or we can have weaker electrophiles, things like carbonyls or alkyl halides, things that have partial charges. And anytime we take a nucleophile and an electrophile, we put them together, we're going to get a reaction. Nucleophile plus electrophile always gives us a reaction. Now, if we look at the two different types of substitution reactions, at least for nucleophilic substitution, anytime we're talking about substitution, it means that we have to replace something. We have to substitute something with something else. So the thing that is attacking we're adding in all of these reactions is our nucleophile. And the entity that leaves, that is replaced, well, we name the leaving group. A leaving group, whenever it leaves, will take with it both of the electrons that it has in a bond. So oftentimes your leaving group will acquire negative charge as it leaves. It is very important that your leaving group be stable in solution. So weak bases make excellent leaving groups. Things like chloride, things like water, weak bases. Fluoride is generally not a good leaving group, and that's just because it's very small. So your best leaving groups will be large, either large atoms or entities that are stabilized by resonance. So just think you want to spread out those electrons over a large surface area, either with big atoms or with resonance, which allows you to delocalize electrons. Now, in an SN2 reaction, I like to think of this as kind of like the home wrecker, where we have a carbon and its X, and this is actually a pretty stable bond. So without the nucleophile present, this bond will continue to exist for pretty much eternity. So in order for this bond to break, we need a really strong nucleophile. I always like to tell my students this SN2 reaction is very similar to the plotline of Titanic, or if you think about this movie, which I know is quite old now by some of our standards, I always think of Rose as our carbon and Cal as the X, the leaving group. Cal was rich, so they certainly probably would have gotten married. And in order to get Rose to leave Cal, you needed that really awesome nucleophile. You needed Jack. So what happens in this reaction is your nucleophile will attack the backside. We call it a backside attack. And it will displace the leaving group all in one step. So here we have, taking 2 to tango, and this makes this a bimolecular substitution reaction, meaning that it is a second order reaction since there are 2 entities involved in our rate determining step, since this is a single step reaction. And this is our concerted backside attack. Now we're going to break down what happens during this reaction and also the different factors to consider that affect the rate of this reaction. The first thing that we may notice just looking at this reaction is that the alkyl halide substrate, when you look at it, kind of has this kind of like an umbrella pointing out. And whenever we have the nucleophile attack, it kind of straightens out and then starts bending the other way. So these reactions are stereospecific, meaning that they only occur in one way. That's because our nucleophile always attacks the backside from the leaving group. And this allows us to maintain a chiral molecule. So if we have like a reactant that's R, our product will be R or S. It will only be one or the other. So the molecule itself is inverted in its configuration. However, based on the priorities of your leaving group and your nucleophile, you may or may not change the designation with R and S. This is another one of the main misconceptions I see a lot of students making. So whenever you're doing, SN2 reactions, make sure that you write out what your product is and then redetermine priorities after the reaction to make your RS designation. So I don't want to see you making that mistake on the exam. So you're going to be glad you came here today. Now, when we look at some of the factors that we can use to predict whether or not something will undergo an SN2 pathway, Of course, we have the kinetics. That's second order, like we discussed. We also want to look at our substrate. Now, remember that the nucleophile needs to come in and attack the backside. I always say when it comes to your molecule, you don't want too much junk in the trunk. Steric hindrance will greatly decrease the reactivity of our substrate in an SN2 pathway. So the less steric hindrance you have, the better the substrate is for an SN2 mechanism. So primary, where you have just hydrogens, is the best. Hydrogens are tiny. You have very little steric hindrance. As you start getting to primary, get a little bit more steric hindrance, but still very doable for an SN2 pathway. Secondary starts to slow down because now you have more R groups, there's more bulkiness. So these reactions will happen, but more slowly. And finally, once you get to tertiaries, there's just too much stuff for the nucleophile to come in and attack. So you will not see tertiary substrates undergo an SN2 pathway. The other thing to consider is the nucleophile. Remember, this is the Jack of Titanic. Jack was played by Leonardo DiCaprio, and if I ever meet Jack, or Leo in real life, I will tell him that he is the best nucleophile ever. He'll never understand what that means, probably, because I don't really foresee him being an organic chemist. But it is true. We need the best nucleophile in order for these reactions to take place. So we're looking for something that has a full negative charge and something that is small. Because if your nucleophile is too big, you're going to have that same issue of steric hindrance. So you're looking for something that's small and negatively charged. The other thing to consider is the leaving group. For all substitution reactions and for all elimination reactions, you always need a good leaving group. There is no exceptions to that. You always need a good leaving group. And remember, leaving group, you're looking for a nice weak base, something that's stable in solution on its own. Finally, we can consider the solvent. Here we're looking at a polar aprotic solvent, something like acetone or DMSO, something that is polar, so it can solvate our either fully charged or partially charged reactants, but it does not form hydrogen bonds. Hydrogen bonds are essentially a way of our solvent interacting with our nucleophile. The way that I like to think about this with that Titanic analogy is if we use something that could hydrogen bond, that would be like literally every person on the Titanic trying to get with Jack. Which I would totally understand that because, I mean, it's Jack, but at the same time, he would never meet Rose. So we want a solvent that is not like hands-on, like something protic, something that can hydrogen bond. We want an aprotic solvent. So that is the SN2 reaction. We're looking for strong nucleophile, something with some negative charge. We're looking for an unhindered substrate. And we will see essentially the molecule turn inside out. Now, that's SN2. An SN1 reaction would be kind of like if instead of casting Kate Winslet as the actress for Rose and Titanic, we cast it with, say, Beyonce, where instead of putting up with all of Cal's crap, she essentially tells him, hey, I want you to pack all of your stuff, put it in a box on the left, and I want you to leave. So here, we are kicking off the leaving group. It is leaving, and we are now like, single ladies. So in an SN1 reaction, our first step is the dissociation of the leaving group. This forms our carbocation intermediate. From here, then the nucleophile can add. So this is actually a two-step mechanism where the rate-determining step is the formation of the carbocation. Because there's only one molecule involved in that step, it is a first order reaction. That's why we call it SN1. So the numbers will, in like SN1, SN2, E1, and E2 will always correspond to the order of the reaction. So if you ever forget, like, oh, is that a first or second order? Look at the number, because that is very telling for our reaction. So this is a two-step mechanism. The stereochemistry here is not conserved as well. And let's think about why that is. Whenever our leaving group leaves, we get this carbocation intermediate. Carbocations are sp2 hybrid, meaning that if you were to look into their geometry, these are flat. They are trigonal planar. So our nucleophile can come in and attack from the top. Our nucleophile could also come in and attack from the bottom, and it does so with equal probability. So in this case, if we do form a chiral product, we would get equal concentrations of both enantiomers. So we call this a racemic mixture. It's not optically active. So stereochemistry is not conserved because of that planar intermediate. Now, if we look at some of the other factors, again, we said this was first order. SN1 is all about our carbocation. The substrate here really depends also on forming the most stable carbocation. I always like to think after you have a breakup, you want to make sure that you have all of your buddies there to stabilize you. So the more R groups you have, the more friends you have, the more stable you'll be as a carbocation. And the rule of thumb here is you always need 2 wingmen. So tertiary is the best, just because you have a lot of R groups, you can really stabilize your carbocation. Secondary will work, but you will not see SN1 for primary and methyl substrates because your carbocation is too reactive to form in solution. So you'll really only see tertiary and secondary substrates. Your nucleophile, however, does not need to be that great. And that's because your carbocation is an awesome electrophile. So it's very reactive, which just means we don't need to be as reactive with our nucleophile. Now, we do need a good leaving group. That's always true of substitution reactions. The last thing to consider is the solvent. Now, one of the most common solvents that you can use for SN1 reactions are actually alcohols or acids. This is kind of like my bad mnemonic to remember this, but I always remember this is the breakup reaction. Everything's about stabilizing the leaving group and the carbocation. That's what alcohol does. So not a good way to remember it, but it is memorable. If we're looking for an SN1 reaction, we want protic. Think about ethanol as our solvent. So just to look at a quick example here, we have a substrate that is very sterically hindered. We have a tertiary substrate. The leaving group leaves, and then we have the nucleophile come in and attack. So it's a 2-step process, and probably what we would be using here for our solvent is likely water. It's also a really great protic solvent. So let's take a look at just a few practice questions here together, and we're really going to illustrate some other key strategies that we look at in our classes. So here we're looking at different substrates as our answer choices. And notice that essentially we have the 4 possible substrates. We have methyl all the way through tertiary. Now we're looking for most suitable for an SN2. So we're looking for essentially an extreme. We know that these reactions tend to kind of follow a general trend going from methyl, primary, secondary, tertiary. So if you forget which one is best for SN2. One of the things I love to tell my students is when in doubt, eliminate the middle answer choices when you're looking for an extreme. So the first thing I would do with this question is eliminate answer choices B and C, because those are the two intermediate answers. I'm looking for the extreme answer. Let's just get rid of them and narrow down our possible list of suspects. Now, SN2, remember, is about that backside attack. Again, we don't want a lot of junk in the trunk. The less sterically hindered substrate is going to be the best. Can't get less sterically hindered than a methyl substrate, so that's what we're going to pick. So again, a little bit of strategy that we can apply here, eliminating half of our answer choices. The other part will come down to content. Let's look at one more question here. Let's take a look at this question. So here we're looking at the rate determining step in an SN1 reaction, and essentially we have a bunch of different steps. Now, what we know about an SN1 reaction is that it is first order. So if you're unsure of what to do, if you forget what this first step is, remember it should involve one thing. So if I look at here, nucleophilic attack on the substrate, that's two things. That cannot be something for a first order reaction. Removal of a hydrogen by a base. Again, you have two things in this reaction. So just by looking at SN1 and narrowing down on the one part, you can eliminate two answer choices. Immediately gone, down to a 50-50. And if you look at this and remember, oh, SN1, that's the carbocation, then you can very confidently go with answer choice C, forming that carbocation intermediate. So when it comes to preparing for your Pcat, what you really want to focus on is both content review and practice. As you've seen, there are multiple ways that the Pcat can test the same pieces of information. So exposing yourself to a lot of practice questions and really mastering strategy is going to be really important for your success, both on the Pcat, in pharmacy school, and beyond. So if you have any questions, continue to reach out to us. Certainly want to make sure that you are in a good place to continue applying these strategies as you continue on with your prep. And if you are interested in learning even more strategies, we didn't talk about like passage strategies, but that's something we do talk about in our classes. So if you're interested in getting even more like down in the weeds with strategy and really mastering your timing of the exam, because timing is always a struggle, definitely reach out to us. We can certainly help you and talk more one-on-one to find the best prep option for your particular needs and test date. So thanks for coming out. Again, if you have more questions, continue to reach out to us. Otherwise, best of luck preparing for your Pcat and your future successful career in pharmacy.